Hello children, welcome to AIMS India's online classes. This is a biology session. You are watching Morphology of Flowering Plants, Parts, Plant Parts and Functions, Part 4. Children, so far we have discussed about morphology. What is morphology? Morphology is the study of various external features of an organism. It may be a plant or animal or any living organism. Okay? The morphology of angiosperms or flowering plants are characterized by the presence of roots, stems, leaves, which are the vegetative parts, flower, fruits and seeds, which are the reproductive parts. Okay, so far we have studied about the vegetative parts such as roots, types of roots, functions of roots, modifications of roots, and the stem, the parts of stem. Okay, that is the leaves. Okay, and their arrangement, that is phyllotaxy, and we have studied about the stem modifications also, that is aerial, subaerial and underground stem modifications. Okay, and leaf modifications we have studied, that is the types of phyllotaxy we have studied, that is alternate, opposite and whorl phyllotaxy. And the modifications of the leaves, leaf tendrils, phyllodes. Okay, and the bladder means utricularia. The leaf is modified into bladder, and pitcher in Nepenthes, insectivorous plant, it is modified into pitcher. Leaf spines in Opuntia, the desert plant, the leaf modified into leaf spines in order to reduce the transpiration rate. Okay, rhizomes, scale leaves in ginger, turmeric. Okay, so flower also we have studied the parts of flower. A flower is a modified shoot meant for sexual reproduction. There are four whorls. What are they? Calyx, corolla, androecium and gynoecium. Calyx consists of sepals, which are green in color. Corolla consists of petals, which are brightly colored, and androecium consisting of stamens, which are the male reproductive part of the flower, and gynoecium consisting of a pistil or carpels. It is the female reproductive part of the flower. Okay, and more we are going to discuss in the today's class. Are you ready, children? Let us proceed. Plant parts and functions. About the flower inflorescence. What is inflorescence? The arrangement and distribution of flowers on the floral axis is called inflorescence. Arrangement of flowers or distribution of flowers on the floral axis is called inflorescence. There are two major types of inflorescence. What are they? Racemose and cymose. Racemose and cymose. There are the types of inflorescence. What is the racemose type of inflorescence? The racemose type of inflorescence is, in this type of inflorescence, the main axis continues to grow. Flowers are borne laterally in acropetal succession. In acropetal succession. In case of acropetal succession, the older flowers are at the base and the younger flowers are at the top. This kind of arrangement is called as acropetal succession. Example, amaranthus you can take. Okay, racemose type of inflorescence. Here the old flowers are at the base and the young flowers are at the top. This is called acropetal succession. Whereas In cymose type of inflorescence, the old flowers are at the top, young flowers are at the base. That is the difference between the racemose and cymose inflorescence. Okay, and some special inflorescence are also present. For example, in Euphorbia, Cyathium, you will study in higher classes. Cyathium is the type of inflorescence. It is a special type of inflorescence. In a verticilla star is another type of inflorescence present in Ocimum. Ocimum means Tulsi. Okay, and Hypanthodium, it is also another type of special type of inflorescence which is present in the ficus. Okay, so cymose type of inflorescence. In this type of inflorescence, the main axis terminates in a flower. Hence, the growth is limited in cymose inflorescence. The flowers are born in basipetal succession or basipetal order. In this case, which means that the older flowers are at the top and younger flowers are at the base. Okay, children, this is the inflorescence, means the arrangement and distribution of flowers on the floral axis is called inflorescence. It is of two types, racemose and cymose. There are further types are there, but only the concept racemose and cymose inflorescence we studied here. Now come to the next part of the plant, that is fruit. Fruit is a mature, ripened ovary which develops after fertilization. Or we can say the mature ripened ovary develops into fruit. The fruit developed from fertilized ovary is called true fruit. Okay, what is a true fruit? The fruit that developed from fertilized ovary is called true fruit, while those fruit that develop from thalamus, the basal part of the flower, thalamus, it is called a false fruit. Okay, fruits are classified as simple fruits, aggregate fruits, or composite fruits. Okay, which you study in higher classes. If a fruit develops without fertilization, it is also very important. If a fruit develops without fertilization, it is called parthenocarpic fruit. Parthenocarpic fruit means seedless fruits. Nowadays we are getting a seedless pomegranate, that is the fruit without fertilization, the fruit which is developed without fertilization. A fruit which is developed after fertilization, it is a true fruit, contains seeds. Okay, whereas parthenocarpic fruits, they are seedless fruits. So this is the cymose inflorescence, where the top consists of old flowers and the base young flowers. It is called basipetal succession. Okay, next, so usually a fruit consists of a fruit wall and seeds. The wall of the fruit is called pericarp. Okay, the wall of the fruit is called pericarp. The pericarp can be dry or fleshy. A pericarp of a fleshy fruit, it is differentiated into three layers. What are they? Epicarp, mesocarp, and endocarp. Outer epicarp, middle mesocarp, and inner endocarp. Okay, a fleshy fruit consisting of the outer layer, that is called pericarp. That pericarp is divided into epicarp, mesocarp, and endocarp. Okay, now come to the seeds. So this is the fruit. Epicarp is the outer layer, which is further divided into middle mesocarp and inner endocarp. So seed is protected by means of these three layers. Okay, means the fruit protects the seed inside. The seed is the ovules. After fertilization, the ovary part is developed into the fruit and the ovule part is developed into seed. Okay, ovules develop into seeds after fertilization. A seed is composed of a seed coat and an embryo. It is made up of seed coat and an embryo. Embryo is composed of two distinct regions. Okay, what are they? A radicle and a plumule. Okay, radicle and a plumule. The radical part of the embryo of seed develops into root system and the plumule part of the embryo of the seed develop into shoot system above the ground. An embryonal axis and one or two cotyledons. Okay, what are the parts of seed means? Seed coat, embryo, cotyledons. These are the three main parts. And embryonal axis consisting of these two cotyledons. Based on the cotyledons, the seeds are divided into monocotyledonous seeds and dicotyledonous seeds. If only one cotyledon is present, then it is called monocot. If two cotyledons are present, it is called dicot. Okay, this is the seed. Seed stores the food material inside, and also contains a baby plant. We are calling it as embryo. Okay, the seed coat, the protective layer, and the first leaves, they are cotyledons. Okay, so based on this number of cotyledons, seeds are classified into two types: dicot seeds and monocot seeds. Okay, before going to that, once again, a seed is a ripened ovule made up of seed coat and an embryo. An embryo is made up of an embryonic axis having plumule and radicle with one or two cotyledons. Okay, if one cotyledon, monocotyledonous seeds, example maize. If two cotyledons, then it is called dicotyledonous seed. Example, you can take most common pea, Bengal gram. Okay, so actually whatever we have studied, the morphology, the total parts which we have studied can be represented symbolically. Okay, that is called floral formula. The symbolic representation of floral characters of a flower is called floral formula. Floral formula, which you will study in higher classes. Okay, now come to the dicot seeds. The outermost covering of seed is called seed coat. The seed coat is composed of two layers. The outer layer is called testa and the inner layer is called tegmen. Okay, seed coat is the protective layer of the seed. So it consisting of two layers, outer testa and inner tegmen. There is a scar. Okay, there is a scar on the seed coat through which the developing seed was attached to the fruit. So this scar is called hilum. Just like in the kidney, hilum is there, no? In the seed also, hilum is present. The hilum is the scar with which the seed is attached to the fruit. There is a small pore above the hilum. It is called micropyle. Okay, in the ovule, there is an opening that is called micropyle. So this the seed also contains the small pore above the hilum, which is called micropyle. The embryo lies within the seed coat and it is composed of embryonal axis and two cotyledons in dicot seeds. Two cotyledons are present in the dicot seeds. The cotyledons are usually fleshy. They are young leaves, we can say. Cotyledons are usually fleshy and contain reserved food materials. They contain reserved food materials. Radicle and plumule, these are present at the two ends of embryonal axis. Okay, embryonal axis consisting of a radicle, that is the root part, and the plumule, it is the shoot part. The endosperm is present in some seeds. If the seed contain endosperm, or the seeds with endosperm are called endospermous seeds. When the endosperm is not present in the mature seeds, the seeds are called non-endospermous seeds or non-endospermic seeds. Okay, here dicot bean seed is given, so it is containing hilum. Okay, what is hilum? The hilum is the spore which is present. Here is hilum is the structure, scare-like structure, which helps in the attachment of seed to the fruit. Okay, and this is the micropyle, the small hole which is present below the above the hilum. There is a small pore is present, that is called as micropyle. Okay, so radicle is the embryonal axis, the part which develops into root system, and this is the plumule in the embryonal axis which develops into shoot system. The seed coat is having a outer testa, inner tegmen. Seed coat consisting of two layers, testa and tegmen. Okay, next so it is consisting of epicotyl, hypocotyl, okay, and the cotyledons are the based on the number of cotyledons. Cotyledons are usually fleshy. If two cotyledons, they are the reserved food material actually. If two cotyledons are present, then it is called as a dicot seed. Here it contains two cotyledons. So this entire structure, plumule, epicotyl, cotyledons, hypocotyl, radicle, this entire internal part, it is called as embryo. Okay, outer hilum, micropyle, testa, tegmen. This is the structure of a seed. Which seed? A dicot seed. Why we call it as a seed? When we will call it as a seed, as a dicot? When two cotyledons are present, then the seed is considered as a dicot seed. Example, bean, pea, okay, pulses, most of the pulses. Next, monocot seed. The structure of monocot seed. The monocotyledonous seeds are usually endospermic, whereas dicot, some of them are endospermic and some of them are non-endospermic. But most of the monocotyledonous seeds are endospermic, but some are non-endospermic also. Example, orchids. Orchid seeds are non-endospermic. In the seeds of cells, the seed coat is membranous and is usually fused with the fruit wall. The endosperm is bulky and stores food. The outer covering of endosperm separates the embryo by a proteinaceous layer. This layer is called alluron layer. Okay, so this is the dicot seed. Once again, comparatively, if you study, you will understand better. So example for monocot, corn seed, okay, maize. So it is an endospermic seed. Endosperm is present. The embryo consisting of same parts, epicotyl, hypocotyl, radicle. Okay, here only one cotyledon is present. Here two cotyledons are present. If two cotyledons are present, then it is a dicot seed. Whereas monocot seed consists of only one cotyledon. Okay, and if you take cells, rice, wheat, maize, so all these seeds are similar. Okay, they contain, the seed coat is membranous and is usually fused with a fruit wall. Endosperm is bulky. That's why when you, if endosperm is present, those seeds when you chew, milky like substance will be secreted in the mouth. That is the endosperm. Okay, It is a reserved or stored food material. The outer covering of endosperm separates the embryo by proteinaceous layer. That's why pulses are proteinaceous. They are separated by a proteinaceous layer. So this layer is called alluron layer. Alluron layer. That's why these pulses, they are highly proteinaceous. Okay. So this is about the structure of monocotyledonous seeds. Okay, children. Let us see more. The embryo of monocot seed. The embryo of a monocot seed is small. Maize seed, this is. It is situated at one end of the endosperm. Okay, the embryo is situated at the one end. See here. It is situated at the one end of the endosperm. It consisting of one large and shield shaped cotyledon. Only one cotyledon is present. Okay. Cotyledon, which is known as scutellum. This is, yes, it is called, it is known as scutellum. There are, so this is the scutellum. There is a short embryonal axis with radicle and plumule. Okay, this is the plumule and the radicle is present below. Okay, the plumule and radicle are enclosed by sheaths. See, these are the sheaths. Enclosed by the sheaths, which are respectively called coleoptiles. They are called coleoptiles and coleorhiza. Coleoptile, the covering of the plumule or stem axis, and coleorhiza is the covering of a root axis, radicle. Okay. See here, the seed coat, micropyle, hilum, cotyledons, plumule is the stem axis, radicle is the root axis. Okay, children, hope you understood. Thank you for watching our video. Please subscribe our channel and press the bell icon for receiving latest updates.